You're all very quiet. Since you're all staring at me quietly, I'll start talking, because it's awkward otherwise. Today we're going to be talking about T-cell development. You can see the slides are here. A couple of other general announcements. So all of the videos are now up to date on YouTube. I posted everything that I was behind on last night. So those are all there. I downloaded all of your stuff to grade, but didn't get as far as grading it. But it's all downloaded and in a folder and ready to go. So that should be something I can do pretty easily. There also, if you recall, and if we look at the schedule, there is a T-cell development problem set due next week, so a week from today, next Friday. That is posted on Moodle. You can also see that I put up the key for the MHC problem set that was due on Wednesday. So all that stuff is posted. The T-cell development problem set, like I said, next Friday. And I think that's all of the things I needed to tell you about. So like I said, today we're talking about T-cell development. We are in some places going to be really thinking about events that are going to sound pretty similar to events you've already heard about in B-cell development. You can see on the board I made a little table of events in B-cell development that we have learned about. So you can see, all of the pro-B cell, the pre-B cell, immature B cell, mature B cell, what the cell's doing, what's on the surface, all of that information sort of about B-cell development, which I will make some references to as we are talking about T-cell development. We talked a little bit last time about the fact that T-cell development happens in the thymus. So we are completely going to be in the thymus for events that we're talking about today. And we are really going to be sort of starting with this cell that has decided that it wants to become a T cell and has moved from the bone marrow to the thymus. So yes, there are some earlier steps where that hematopoietic stem cell has to decide if it's going to be a myeloid cell or a lymphoid cell. There's a step where it sort of has to decide B cell or T cell. We're starting with the cell having already decided T cell and heading to the thymus with the events that we're going to be talking about today. Yep, Jazz.
So it starts off as the common T cell /
ILC, yep.
ILC precursor in the thymus.
For our purposes, yes.
Okay.
It might actually be here, to be honest with you, that it's in the thymus. We might be at this common T-cell precursor. As I think about it, I only know about one of these types of cells very well. And that one doesn't come from the thymus. And so maybe not, but I'm not positive about the other ones. We're not gonna get super into them right now. So this is sort of the process we're going to think about. So if we wanted to be super simplistic about T-cell development, I will show you how we would describe T-cell development in a super simplistic way. So what you can see in my very simplistic way of talking about T-cell development is that I took my table that described B-cell development and I erased the B's where I said B-cell or B-cell receptor, and I wrote T's for T-cell or T-cell receptor. And big picture, that's what we're going to be seeing with T cell development as we talk through the details of T cell development. Unfortunately for us, the T cell field uses some slightly different nomenclature. And so we're going to have to think about some slightly different nomenclature for some of these same steps. The other things that are going to sort of make T cell development a little tricky compared to what we saw with B cell development is we have to deal with that whole alpha, beta, gamma, delta thing that we saw last time. And the testing process with T cells is a bit more involved. And so those are the places where we're going to be seeing slightly different details. And so T cell biologists don't talk quite as much about pro T cell and pre T cell. You don't hear those names as often. We hear a different set of names for T cells undergoing different parts of T cell development. And that is because when we started to understand T cells undergoing T cell development, we had flow cytometry. And so we could tell that there were certain types of proteins on the surface of cells at different stages of T cell development. And so the people who were studying T cell development were doing a lot of flow cytometry to break down the developing T cells into different groups. The first type of thing that those people who were looking at T cell development looked at was they looked at all of the developing T cells in the thymus and their expression of CD4 and CD8 on the surface of that cell. And when they did that, they saw that developing T cells in the thymus fell into 4 groups. One group that had no CD4 and no CD8, so they were CD4 minus, CD8 minus. They were down here in this lower left quadrant. We've got one group that is CD8 positive, CD4 negative, which you can see is in my lower right quadrant. CD4 positive, CD8 negative, up in this upper left quadrant, and then CD4 positive, CD8 positive, which are called double positives, which are in the upper right corner. And these were named, so when you're talking about developing T cells, oftentimes you will hear about double negatives or DNs, double positives or DPs, or the CD4 single positives or the CD8 single positives. And so we often will look at T cell development and talk about different cells that are at different stages of T cell development as things like double negatives, double positives, or either of our 2 types of single positives. You can see an actual flow cytometry plot of the thymus here. And I will also draw a version of this same plot where we have the CD8s and the CD4s. If you look at the percentages of cells in each quadrant, you will see that the lowest percentage is the double negative cells, which are shown here as 3%. And so if you were going to draw this plot for me, one of the things that I would be looking for is that the double negative population would be the smallest. And so I would draw a little tiny circle there. If you look at the double positive population, you can see that it is the largest in the thymus. And so again, if you were drawing this flow plot for me, I would expect that to be the biggest population. I would expect a big circle. And then you can see that the 2 single positives are in between. They're not as small as the double negatives. They're not as big as the double positives. And so I would kind of want those to be like in between size circles. I don't care. Technically, as you can see here, they are not the same size. I'm not going to like measure with my millimeter ruler to see if they're exactly right. I just want this one to be smallest, this one to be biggest, and the other two to be somewhere in between. And what we have learned in studying T cell development is that developing T cells at the beginning of their time are double negative T cells. And then during the process of T cell development, those double negatives will become double positive. You can even see I've got a little arrow. The double negatives become double positives. So double negative is the earliest T cell in development. Double positive comes next. And then the cells sort of branch out to either become CD4 or CD8. And you can see those arrows branching out here. So double negatives are first, double positives are second, and then either type of single positive is third. When people did this work, they also found out that the cells in some of these groups are not a homogeneous group of cells. So you can further subdivide some of these cells. And in particular, we further subdivide the double negatives. So we don't just have double negative, double positive, single positive. We actually have different kinds of double negatives. So this is an example where you might make this plot of CD4s and CD8s. You might gate on the double negatives, basically just double click on the double negatives and say, I want to look at these cells with other markers to sort of figure out which group of double negatives my cells are in. People get a little confused when I show this here. Just know that this image and then this other image that I have, which is actually dividing up double negatives, are not from the same source. Everyone's like, but there's not that many cells here and there's lots of cells here. Yeah, they're different experiments. Like, yes, I do not have a good image of like the exact same plot where you do the thing. So yes, there's not that many cells in that one, and they had to do a bunch of work to get a lot of cells in this one. It's okay. And so what we now know is that if we can look at the double negative cells using CD25 on one axis and CD44 on the other axis. I will probably mention CD25 later on in a few lectures. We may hear about CD25 again. CD25 is actually pretty important. While in my life I have spent lots of time thinking about the importance of CD44, you don't have to care about CD44. You just have to know CD44 is one of the things we use to divide up our double negatives into subgroups. And so one subgroup is the CD44 positive, CD25 negative population of double negatives. Those are known as DN1s. You can see DN1 is labeled here. DN1s are the first cells that come into the thymus. They're the earliest ones in T cell development. So that's why they get called DN1. And you can see that the DN1s are CD44 plus, CD25 minus. After the cell is a DN1, the next thing that it's going to do is become a DN2. And a DN2 is CD44 plus, CD25 plus. You can see that here. Then the cell is going to become a DN3. You can see the DN3s here. And so the DN3s are CD25 plus, CD44 minus. After being a DN3, this cell is going to become a DN4, which is CD25 minus, CD44 minus. After the cell is a DN4, it becomes a double positive. So these 4 steps are like sub-steps within being a double negative. Once you finish those 4, then you go to being a double positive. And so this is how a lot of T cell biologists often describe aspects of T cell development. One thing that is sort of nice here I don't have it written out perfectly in my table that I've written on the board, is that if you remember when we talked about our B cell development, and we talked about pro-B cells versus pre-B cells, in some cases in the book and in our thinking, we kind of said, well, they do a couple things in an order. Like we said, in your textbook, sometimes they talked about early pro-B cells versus late pro-B cells. Like the early pro-B cell had to put together D of the heavy chain and J of the heavy chain, and then later it did V to DJ. And then when we got to the pre-B cell, first it had to put its receptor up on the cell surface, and first it would survive and proliferate, and then after it would turn off RAG during that time, and then it would turn RAG back on so that it could do light chain rearrangement. So again, we were kind of thinking about like an early versus a late pre-B cell. The nice thing about this division into double negatives versus double positives and the subtypes of double negatives is that sort of some of those different events actually get their own name for a step. It's not perfect, but that's sort of the real benefit here. So if we think about T cell development, we can first think about what's happening in our double negatives. And so we'll start with double negative 1, DN1, and DN2, which you can see here. And DN1 and DN2 are really the cells that are sort of the equivalent of a pro-T cell. DN1s are the cells that are just getting to the thymus. So DN1s are sort of the early ones who just made it to the thymus. And then DN2s are the cells that are actually starting to do rearrangement. So they're starting to do that rearrangement here. Again, with T cells, life gets a little more complicated because we have two kinds of heavy chains and two kinds of light chains. Remember, we have this choice that the T cell has to make about being an alpha beta versus a gamma delta. If it's an alpha beta, we've got beta as the heavy chain. If it's a gamma delta, we've got delta as the heavy chain. And so what you will notice here is that it says on this slide, and we will say this a few times, we can see that the beta rearrangement is happening. So beta is sort of our normal alpha beta T cell heavy chain. So you can kind of think of that as we're doing heavy chain, just like we were doing before. It turns out the T cell also does gamma and delta at this time. So it rearranges delta, rearranges beta, rearranges gamma, all at this time. So the normal heavy chain beta, as well as both the two weird chains, are getting rearranged at this time. These events are happening in the cortex. So you can see the T cell is coming into the thymus, kind of right at the border between the cortex and the medulla. And then that T cell is moving into the cortex and starting to do these rearrangement events. According to this slide, Jazz, it looks like they can still give rise to NK cells and the other cell types, and so apparently the answer to your question is yes. So you can see that we've got our DN1, DN2s doing that rearrangement, and they're now in the cortex. They came in right at the border between the cortex and the medulla. First, let's think a little bit about what happens if the T cell is successful in making its beta chain. So first, let's just think about, let's imagine successful beta chain. This is kind of like the simple version of what's going on. This is kind of like the parallel to what we saw with B cells. Sort of like successful heavy chain, hooray. Now what we want to do is we want to test that heavy chain on the surface of the cell. And so we will put that cell with its successful heavy chain of beta on the surface of the cell as part of a pre-T cell receptor. And at this point, our cell will be a pre-T cell. And so you can see once we get to sort of DN3, we are now a pre-T cell. We now have our receptor on the surface of the cell. Perhaps we get a good signal through that, and we're able to sort of continue through the development process, just like we saw with B cell development. And you can see that DN3 has a pre-T cell receptor on its surface, where we have that beta heavy chain along with some other light chain, because we haven't made light chain yet. We have to have our surrogate light chain. And so we need surrogate light chain. So this isn't a T cell receptor yet. This is a pre-T cell receptor. It only has the beta heavy chain. It doesn't have a real light chain yet. It has this surrogate light chain. And this surrogate light chain is called PT alpha. I've also sometimes actually heard it called pre-T alpha. I think I personally just use those two terms interchangeably as PT alpha and pre-T alpha. But you can see that we've got this surrogate light chain, PT alpha, that's going to pair with the beta chain, be on the surface of the DN3, and allow for testing of the heavy chain with some kind of signal. We know that this cell doesn't have CD4 or CD8 because it's a DN. DN means double negative, means no negative for CD4, negative for CD8. But this cell does have the CD3 chains at this point on its surface to allow for signaling through the beta chain and to allow the cell to get its pat on the head to say, you made a good heavy chain. Good job. Jazz, did you have a question?
So in this instance, it's the PTA because it's a beta heavy chain?
Yeah, right here we're talking about what's happening with beta. We haven't gotten to what's going to happen with the other ones yet.
Okay, thank you.
Yep. Just like with the pre-B cell receptor, there is a question of how the heck do we get signaling from the pre-T cell receptor, because this thing doesn't have all its parts yet, so it can't bind antigen. It can't be specific yet for antigen. And we're pretty sure that the way that the pre-TCR signals is that two adjacent pre-TCRs can bind together. So the beta of one can bind to PT alpha of the other, and they can sort of all get pulled together. They can sort of get that induced proximity just by getting sort of linked together kind of non-specifically, and we can get this signal transduced in this cell. So basically, as long as you have an actual functional beta chain, you didn't put in a stop codon and make a protein that's too short, you got a legit protein, you're going to be able to make this sort of big structure on the cell surface and get some kind of signal. And when that cell gets that signal, we're going to have a bunch of events happen, just like we had in the B cell. So the first thing that we need to do is we need to stop additional beta rearrangements. We got a good beta chain. We don't want to make a second one. We want to have allelic exclusion and end up with a T cell with only one T cell receptor that has specificity for only one MHC plus peptide. If it didn't do this, we would have disaster of our T cell responding to two things. That would be very messy. We don't want that. So we are going to turn off RAG. We're going to stop our rearrangements at beta. We're going to do allelic exclusion. We're going to get proliferation. It's key that that proliferation is happening while RAG is off, so we don't see any DNA damage. We're going to tell that cell to survive. Good job, you should live. And not only should you live, you should make many progeny in proliferation, so we can take your successfully made beta chain and pair it with many alpha chains. If you have many progeny, they could each make a different alpha chain. We can get lots of pairs of that good beta chain with some alpha chain. So we're going to stop our rearrangements. We're going to survive. We're going to proliferate. We're going to stop making PT alpha. We're going to stop transcribing PT alpha because we don't need it anymore. We used it. We are past the point in our life where we need that. We are also potentially going to, we are now going to start transcribing and translating CD4 and CD8. And again, it's both CD4 and CD8. So when we get to the point where the cell is transcribing CD4 and CD8, it becomes a double positive. Double positive means you have CD4 and CD8. And so many of these earlier steps, the allelic exclusion, the proliferation, the survival, the turning off transcription are sort of our DN3 to DN4. Turning on expression of CD4 and CD8 will sort of mark when you become a double positive. Once you actually make CD4 and CD8, you're now positive for CD4 and CD8. So you're a double positive. And you're also going to start then rearranging alpha. And so what you can see is that for the pre-T cell is actually a bunch of things. A pre-T cell is a DN3, a DN4, and a double positive. And it's really about which of these different events are happening in the cell at which time. All of these events that we are talking about are happening in the cortex. And so you can see that our DN2, DN3, DN4, all are different types of interactions that are happening in the cortex. We have not yet moved into the medulla. And again, one thing that you can notice in that previous slide that I just showed you is that one of the events that is happening in our DN3, DN4 is proliferation. We're getting a lot of cell division here. And if you remember when I showed you the image of the thymus, the cortex is the one that's super jam-packed with cells. It's because that's where the proliferation is happening. And you can see that proliferation happening here as one of the events that happens when we get a successful beta chain. This slide also sort of mentions in a couple places TECs. TECs are thymic epithelial cells. You can also think about the thymic epithelial cells as structural cells of the thymus. You can also think of them as the thymic stroma that I mentioned to you last time. So I told you about the thymic stroma, and I sort of was saying, you know, the structure parts of the thymus. A lot of the slides today mention TEX, and TEX, the structural cells in the thymus and the stroma, are interchangeable as far as you're concerned. When I was editing some of those videos last night and I was posting them, I was like, man, why didn't I actually say that in a past lecture? That would have made everyone's life easier. But I didn't, but I'm saying it now. So all this is great, but we have this question that sort of Jazz brings up about, well, what about gamma and delta? We've talked about beta rearranging, but I told you at that step where beta is rearranging with the pro T cell, we also see delta and gamma rearranging. And if you look at this image, you can see that the cell could, at a couple spots, branch off and be like, yeah, I'm not going to do the whole alpha beta thing. Instead, I'm going to become a gamma delta. And so at some point, the cell could sort of leave the path of double negative, double positive, etc., of normal alpha beta T cell development and decide, yeah, I'm going to go for this double negative or this gamma delta route. Once it's a gamma delta, all of these things like double negatives and double positives and all of that stuff doesn't really apply to it anymore. It just gets to be a gamma delta. And so this is how the cell kind of deals with and sort of thinks about being a gamma and being a alpha beta versus a gamma delta. As I told you, at that first step in the pro T cell, the cell is actually doing rearrangement of beta, gamma, and delta all at the same time. And really, it's a race. If the cell gets a successful gamma and a successful delta first, those end up on the cell surface and they allelically exclude further beta, gamma, and delta rearrangements. If the cell successfully gets a beta first, then the beta ends up on the cell surface and allelically excludes further beta, gamma, and delta. And so basically it's about all these loci are sort of trying to do the correct recombination. In the case of gamma and delta, you got to get 2 right. So that's a little bit of a challenge. But in the case of beta, you got to get 1 all the way right. So until you get a beta or until you get both a gamma and a delta, all this VDJ recombination at all of these loci is going on. And then when you're successful, either with a beta or with a gamma and a delta, then you can get a functional protein on the surface of the cell. And that functional protein will give a signal that leads to the allelic exclusion and stops further rearrangements of gammas, deltas, and betas. So maybe you finish beta, you are trying to put it on the cell surface, but it sucks. And so you don't get a stop signal. You don't get told stop because that beta didn't actually work. It didn't actually have a full protein because it got a stop codon somewhere in the middle. So you finish a beta, but but that beta sucks, so you're gonna have to make another beta. And while all that's happening, you're also doing gamma and delta, and they get done first. That's kind of how it works. While if you kind of do a beta and it's right on the right try, on the first try, then that's gonna go up and that's gonna further allelically exclude out the gamma and the delta. Yep, Paige.
So a gamma delta T cell it's not ever T cell?
So yeah, once it's a gamma delta T cell, it's just a gamma delta and is down its own life path. Yes, Jazz?
I know it's technically pretty much just one, but what's the proportion of delta gamma to alpha beta? Is it theoretically 2 to 1?
Don't know the answer off the top of my head. And you'd have to also think about with that question, you could imagine, and this is I think why it would be tricky to do, and also why I don't read that literature, so I don't know the number off the top of my head. You could also look at what's the number in terms of like T cells developing in the thymus versus those that get out, right? You could, like, do you measure in the periphery? What's the percentage in terms of how many got out? Because we haven't had any steps where we did any like testing and killing yet. And so the answer might be one thing like right now, but different when they actually get out into the periphery. Or maybe they proliferate at different rates in the periphery. And so then that also would influence it. So really, to answer your question, you'd have to actually like look at a thymus and find the cells at a specific step of development and then put them into gamma delta versus alpha beta. I don't know a paper that's done that, but again, I also don't read this literature as closely as some other literature. Yep.
So they all start as double negative, but then once you become a gamma delta, that sort of like terminology stops.
Yeah, you just go off on your own gamma delta road. Yep, Chrissy.
And so when gamma and delta both need to be ready, it's not just like one of them?
Yeah, you got to have both gamma and delta ready. So if you can't say put delta with pre-T alpha or something like that, because delta is the heavy chain. You got to have delta can only go with gamma.
Okay, so they both need to be ready, and for beta,
You just need beta, yeah. And so you can sort of see this process here. This, I think, answers some of your questions about, the road. You can see here's our cell. It's sort of trying to make beta, it's trying to make gamma, it's trying to make delta. If it successfully gets a beta, then it continues being a DN and a DP and all of that stuff. If it makes gamma delta, then it becomes a gamma delta and just goes down like the gamma delta life path. And so you can see that happening here. You can also, as I'll also show you, and so actually, gamma delta cells don't ever have CD4 and CD8 on their surface. They're like completely weird T cells. Whenever I talk about CD4s or CD8s, and honestly, whenever I talk about T cells, I'm talking about alpha betas, which are sort of the standard ones. We think that basically the way that this also works is that the signals that happen downstream of beta and PT alpha are a little bit different than the signals that come from gamma delta in the thymus. It may be, for example, how weak or strong that signal is that the T cell gets, because whatever that signal is that the T cell gets is going to basically change that T cell's life. It's going to tell the T cell what set of events are you going to do for the rest of your life? What kind of functions are you going to do for the rest of your life? This sort of decision point really is going to dictate the rest of the life of what that T cell is going to do and start like a specific program of like, you're going to do this kind of functions versus you're going to do this kind of functions. And so we think that's all based on slightly different strength of signal that's coming into the T cell at this point during T cell development. If you look at this image, you can also see that the cell can still make that decision to go to be gamma delta a little bit later as well. So if I go back to this slide, you can see that some cells decide to be gamma deltas sort of pretty early off with DN2. Maybe that's because they made a gamma and a delta first and they looked great. You can also see that some cells make the choice to become a delta gamma a little later, or a gamma delta a little later. Apparently I'm talking about sororities instead of T cells now. You can see that this can also happen a little bit later. And basically when this happens a little bit later, we're seeing a situation where the cell is not being able to make a good alpha. So maybe it made a good beta, and it try as it might, it can't make an alpha. It just keeps messing up alpha. At some point, it might actually get gamma and delta rearranged. And at some point, like, it's better to become a gamma delta than to be a beta and die because you can't make an alpha. So sort of even if you make a successful beta, if you stay as a pre-T cell sort of with a successful beta and just never really get a successful alpha, you can still end up going down the gamma delta route. Yep, Chrissy.
Is that like it definitely happens if it can't make a good alpha?
It is an option that could happen if it can't make a good alpha. The point of telling you this is that we can talk about sort of what is the point of no return. When is the cell like, I definitely am alpha beta, I am no way going to do gamma delta, like that option is closed to me. Where is the point of no return is kind of where I'm trying to get us to. And the point of no return is when you make alpha. Because when you make alpha, you actually delete delta. So when you make alpha, you actually have to throw away the delta DNA. And so, because remember, the entire delta heavy chain locus is in between V alpha and J alpha. And so once you make a good alpha, you have thrown away delta, and you have thrown away your chance at gamma delta life. Now you are absolutely committed to alpha beta life, and you have to do alpha beta because you threw away delta. So if you have a beta and you really kind of don't get there with alpha, you could still go down the gamma delta path. Once you actually make an alpha, you're done. Yep, Paige.
How do you test an alpha without making it? Like, how do you know it's not going to be successful without making it?
Well, so remember you got 2 chromosomes. So maybe you make it on, maybe you do one, it's not successful. You're kind of sitting around as a pre-T cell for too long and the cell's like, fine. Okay?
Yeah.
And so these are the events that are happening in our DN3, our DN4, and then eventually into our double positive. So again, remember that I showed you that with our cell that is doing, that has a pre-T cell receptor, it sort of gets, when it gets that beta chain on its surface, it is a DN3. But there are lots of events that happen here. So this is a pre-T cell at DN3. But there are lots of events that happen. And so all those pre-T cell events are happening while the cell is either DN3, DN4, or double positive. So again, we had first this allelic exclusion. We've got some proliferation. We've got some survival. We've got some turning off pre-T alpha transcription. All of those things are in the DN3, DN4. Then we turn on CD4 and CD8. Once CD4 and CD8 are on, we're a double positive. And then we can finally start doing our alpha chain rearrangement. So in our double positive cell, basically we're doing alpha rearrangement sort of as we're becoming a double positive. I honestly always learned that alpha rearrangement happens on this arrow between double negative and double positive, between DN4 and DP, that alpha rearrangement sort of happening right there. So we're going to do our alpha rearrangement, and we can imagine that we have this double positive cell that now has an alpha beta T cell receptor. So it's got a T cell receptor, and it's got CD4, and it's got CD8. And so you can see this double positive here. And this double positive is basically our immature T cell. If you remember when we talked about B cells, that cell that has finished making a heavy chain and a light chain, that immature cell, is now ready to test for self-reactivity. And so again, we're going to need to test for self-reactivity with this cell before we let that cell leave the thymus and go to the periphery. We also are going to have that cell choose CD4 or CD8. So one thing to note is that this plot that I've showed you, this CD4, CD8 plot, has this huge population of double positive cells that have both CD4 and CD8. Those double positive cells, cells that have both CD4 and CD8, only exist in the thymus. If I look anywhere in the periphery, I never find a CD4, CD8 double positive cell. Those are only cells that exist in the thymus. And so the cell kind of like has to make a choice. Am I going to be CD4 or am I going to be CD8 before it leaves? We're only going to talk a little bit about that. We're going to talk a lot about the testing for self-reactivity. But both of those decisions have to happen before the cell leaves the thymus. And so this really gets us to thinking about central tolerance in T cells. And so again, we talked about central tolerance in B cells. So once our B cell had made a heavy chain and a light chain, it got a little bit of a signal. Then it had to be tested before it could leave the bone marrow. And we saw that central tolerance process. When we talked about B cells, there were 3 choices that could happen to the B cell. The B cell could either get deleted, that B cell could undergo receptor editing, or that B cell could undergo anergy. There are going to be slightly different choices for the T cell. When we talked about B cell development, we also talked about the fact that there was a little bit of a flaw with the plan. There was something that you might think about that say, that kind of sets us up for the possibility of autoimmune disease. And that thing that we talked about, we said, it's a good thing that peripheral tolerance exists, because this is a way that stuff could slip through the cracks in B cells. So what was the issue with B cells and B cell development? Yep, Julie.
Wasn't it that it only tests the self-antigens in the bone marrow and not like the other antigens that could be in the body?
Yep. So the antigens that are present in the bone marrow are only proteins made in the bone marrow, or carbohydrates or lipids or whatever made in the bone marrow. And the bone marrow doesn't have every protein in the body. It doesn't have the eye-specific proteins and the brain-specific proteins and things like that. And so the developing B cells get tested against like antigens that are everywhere in your body, or like ones that are in the bone marrow, but they don't get tested against like ones that are in specific tissues. And so you could imagine you might have a B cell that responds to, say, myelin in the brain. That B cell is not going to see myelin in the brain in myelin in the bone marrow. It's not going to get tested. It's not going to get deleted. And so it's going to be an autoreactive B cell that's going to leave the bone marrow. And we said, thank goodness for peripheral tolerance. I point that out because, again, we're going to do some comparisons with what we see in T cell development. And sometimes when people learn sort of how this works in T cells, they really don't like T cells because they're like, wait, what? They're like, so basically, like, I'm definitely getting an autoimmune disease. Like, sometimes people leave this lecture with, like, the message of, how am I alive? Because they feel like the issue with T cells is so big that you're like, no, that's bad. That's real, like, no, that can't work. And my answer for you is, thank goodness for peripheral tolerance. So yes, you are alive. No, you are not, this the details I'm going to tell you about may seem like scary and catastrophic, and we are glad that peripheral tolerance helps us deal with them. So just remember that. Don't feel totally freaked out when we talk about this. And this is because when T cells are doing their testing in the thymus, they actually have to really test two things. There are actually two things we got to figure out if you're a T cell in the thymus. One thing, obviously, you got to figure out is, am I self-reactive, right? We need to test for self-reactivity in the thymus and get rid of the things that are reacting to self-antigen. But we also have another thing we actually have to test for in the thymus. The other thing we have to test for in the thymus is, does my T cell receptor recognize self-MHC? So remember that I told you last time that developing T cells learn what self is from the thymus. And so basically, your T cell receptor could bind to any old thing. We want it to be able to bind to self-MHC in order to get signals. If you had a bunch of T cells that came out of your thymus and didn't bind to self-MHC, those would kind of be useless, right? So you got to make sure that the T cell binds to self-MHC before it's allowed out of the thymus so that it's useful. So we have to be checking two different things. Are you self-reactive? We don't want you to be self-reactive. That would be bad. Can you bind to self-MHC? If you can't bind to self-MHC, then you are useless to us. And I don't want to give my glucose and my ATP and stuff to useless cells. I would like to save all my resources for the useful cells. So we have to kind of think about both of these things. So there's two sides in T cell development. And so you can see that our double positive T cell is kind of thinking about two different kinds of signals going on. So like I said, we got two boxes we're trying to check. We got two signals, two things we need the cell to figure out. Get ready. This is where everyone starts to be angry. There's only one receptor that's trying to test two things. So somehow we got to get two tests off of one receptor. And there's really only one kind of antigen in the thymus. So it's not like we can go, I'm going to test here for self, and I'm going to test here for MHC. We only got one receptor. We're kind of doing all of this at the same time. So we talk about it as separated, and there is a little bit of separation time-wise, but really it's one test that the cell is doing. And we can think about this test that these cells are doing with a model that immunologists talk about all the time, which is known as the Goldilocks model of T cell development. So we've got our developing T cell. Let's imagine our developing T cell gets a really, 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 really strong signal. It's in the thymus. It binds to something. It's like, this is the best. I get so much signal from this. Or imagine it gets like a good amount of signal from a lot of things. Basically, it's getting a high cumulative signal. It's getting a ton of signal in the thymus. What's up with that cell? I'm continuing my hug on purpose. Yeah, Emily.
I would say self-reactive.
That self is probably self-reactive. If it binds to one thing crazy hard, that's self-reactive. If it's binding pretty well to a lot of things, that's probably pretty self-reactive. Do you want that cell? No. And so what we're going to do to that cell is that we are going to actively kill that cell. We are going to make that cell undergo apoptosis. And so this is going to be a strong signal. I think in some places I list it as high, medium, low. So if we're doing high, medium, low, this is high signal. If our developing T cell gets a high signal at this point, that developing T cell is going to undergo apoptosis. We're basically going to actively kill that cell. Sometimes we refer to this process as active killing as negative selection. So we are going to negatively select our T cells if it is self-reactive. So if it has a high signal in the thymus, that is bad, bad, bad. We are going to negatively select it, which is basically means we're going to delete it. Sometimes in like a, I don't know, a metaphor-y kind of way, immunologists in some cases will also refer to this as killing the harmful. So we're going to kill the harmful cell. We're getting rid of that cell that's getting way too much signal in the thymus. And you can see, in fact, that might be about 2 to 5% of all the developing T cells have that problem. But now let's imagine the other side of this. Let's imagine that you are a T cell who has made a T cell receptor that binds to nothing in the thymus. You get absolutely no signal in the thymus because you bind nothing. You get no pats on the head from your T cell receptor because your T cell receptor doesn't bind anything at all in the thymus. What does that mean? Yeah, Jazz.
You have no adaptive immune response.
Well, maybe, but we're just talking about an individual T cell. We're not going to talk about globally. If you as an individual T cell bind nothing, are getting no signal in the thymus, what does that mean, Miles? It doesn't get a signal to survive. Yep. It doesn't bind to your MHC. It can't bind your MHC. It gets nothing from your MHC. You need the cell to be able to get a little some pat on the head from your MHC, right? If that cell gets nothing from your MHC, it's not a good T cell. It's not a T cell you actually want to put out into the periphery because it's useless, right? And so we're going to have some cells that get kind of like what I'll call a medium signal or an intermediate signal. That cell is going to get the signal to survive. That's going to be our pat on the head, right? And we're going to get some that are going to get like a really low or like no signal. They're useless. They get no pat on the head. And so we call these cells that survive, because they get like the medium signal, the intermediate signal, we call this positive selection. This cell got told, yeah, you bind to self-MHC. You were positively selected. You get a pat on the head. But then there are some other cells that can't even bind to self-MHC. They get no pat on the head. Those cells are referred to as failing positive selection. So those cells fail positive selection. They have failed to get a pat on the head. Do we want these cells to go into the periphery? No, they're not helpful at all. They are so unhelpful, they're not even worth our effort to kill them. What we actually do to these cells is these cells they do end up dying by apoptosis. But this apoptosis is a little bit different than what you saw on the other side. It's not quite the same as what you saw with the strong signal. These cells undergo what is known as death by neglect. And so basically, these cells never get a pat on the head, and so they die. And so you can see these cells will undergo death by neglect because they don't even bind to self-MHC in the thymus. And so if we have low or no signal, we have death by neglect. It does end up being apoptosis, but sometimes people just say apoptosis happens to various cells, and I want you to realize this is different. This was like we neglected the cell and it didn't get any signal and it died, not we actively killed it. In my little metaphor thing, where we talk about negative selection as being kill the harmful, when we talk about failing positive selection, we say we ignore the useless. Yes, I'm being so mean to T cells, I know. You can notice that more than 90% of T cells have this happen to them. And so again, you have to, in order to get T cells that actually are in your blood, you have to make a ton of T cells because you're gonna get rid of some of them. When we get the combination of these two things, you can see that it's way over 95% of T cells are dying. And so this one textbook that tries to be funny about immunology that I have calls the thymus murder university because of all of this cell death that's going on. And so you can see if we have our cell that has a really high affinity for antigen in the thymus, it's going to get deleted. You can also see from these images and from my way of writing this, there is sort of this fourth thing of what happens if you have sort of not all the way strong, but sort of medium strong. So basically what you can see is I have no signal, a low intermediate signal, a high intermediate signal, and a high signal. So you can, and here they're actually showing you low, intermediate, and high, and they're not even showing you none. Here they're showing you this fourth one as high as opposed to very high. So you can see that there is basically this other option. There is another option. I'm going to tell you what the other option is later. Today you are not going to learn what the other option is. But at some point I'm going to be like, oh yeah, remember that other option? This is the other option I'm talking about. So then we've got to think about these sort of medium cells, these cells that, in the case of Goldilocks, because again, this is called the Goldilocks model of thymus selection, they didn't get too much signal. See, these ones, too much signal, too much like Goldilocks. These ones, too little, not enough like Goldilocks. So we've also got this group in the middle who got just right signal, the intermediate amount. They got a pat on the head, but they didn't get a pat on the head that was super strong. Those cells are positively selected. And those cells are allowed to leave the thymus as your T cells and to go be your T cell in the periphery. So those cells survive. They are positively selected. And we keep the useful. And so you can see that it is a relatively small percentage of T cells that are the useful T cells that get the positive selection, they get the good pat on the head, and they get told that they can leave the thymus. And so you can see here sort of this positive selection here. If our cell gets absolutely no signal, it dies. So here you can see death by neglect where we're getting absolutely no signal. And here you can see the cell gets sort of moderate signal, it's going to live. And so that cell is positively selected. This is how our developing T cells find out what MHC is self. This is why a developing T cell sees the MHC of the thymus as self. And really, these developing T cells are seeing the thymic epithelial cells, the structural cells or the stroma of the thymus. They have MHC on their surface. The developing T cells are checking that MHC, and they're basically learning from that check, this is self. Only the ones who interact with that are positively selected and are able to leave the thymus to become T cells. And then you can also see negative selection happening. So you can see these events of negative selection here, where again, if the binding's sort of medium, the cell is living. If the binding is really high, the cell's dying and is getting negatively selected. In general, many of these events are happening now as we're getting to the medulla. So a lot of this cell death is happening in the medulla. Particularly the negative selection is happening in the medulla. If you get a really large signal in the medulla, that's negative selection signal. And that's why the medulla has way fewer cells in it, because the cells are undergoing cell death a lot in the medulla because they are either being neglected or they are undergoing negative selection. What you can also notice, I erased it. If you think about our plot of T cells in the thymus, the double positives is the biggest population. That's because that's right when the proliferation has just happened. But before the cells have to make that choice to become single positive, they might go through death. So we get proliferation, we get this giant double positive group, but before we get to being double negatives, some of those cells are going to die. And so the double negative populations are way small, or the single positive populations are way smaller. The double negative population is way smaller in this population because of proliferation. So we need to think about a positive thing and a negative thing about what's going on with T cell development. So I'm going to tell you about this sort of cool thing about T cell development, and then we're going to come back and learn about the catastrophic part about T cell development. I'm going to end up probably leaving you on that cliffhanger. So I told you that with B cells and B cell development, we have this problem. And the problem is that every antigen is not present in the bone marrow. So we can't test developing B cells against every antigen. It turns out the thymus is special. And, before I tell you that the thymus is special, the other thing I want to tell you about the thymus being special is that epithelial cells in the thymus present on class 2. So good news, all cells of the thymus have a nucleus, so they all present on class 1. So we can definitely show our developing T cells what class 1 looks like and have class 1 be a thing that we can test for and say, do you recognize class 1 in T cell development? But thymic epithelial cells, those structural cells of the thymus, also express class 2. So the thymus is also special in that it's making class 2, and we can also test our developing T cells. Do you respond to class 2? Do you get a signal from class 2? So that's one thing that's special about the thymus, is we got class 1 and class 2. So there is sort of this special thing where, the thymic epithelial cells, the TECs, do have class 2 so that they can support T cell development and testing of those T cells. But for a long time, we also wondered a lot about what was going on with tissue-specific antigens. And so this image is showing you 2 tissue-specific antigens. One's in the retina, one's in the ovaries. So it's just a protein that's only made in the retina and a protein that's only made in the ovaries. And if you remember with B cells, this was a problem because the B cells didn't get to test their B cell receptor against them. It turns out the thymus is special and that the thymus makes every protein in the body. So every protein is made in the thymus, so you get to test developing T cells for self-reactivity against everything. So those cells, those proteins that are in the retina and also in the thymus. And so we can actually test against every protein in the body in the thymus because the thymus is special and makes every single protein. And so the thymus is really great in that it is helping us with this process of negative selection. For a long time, we didn't really know how this worked. But people actually later found a particular gene called AIR. And AIR is a transcription factor that acts in the thymus that transcribes everything. That is the simple version of AIR. There is a much more complicated version of AIR, but we're going to go with that. AIR basically supports the transcription of everything in the body. AIR makes sure that the thymus makes every single protein in the body. Technically, it's not a transcription factor, but that's okay. Yeah, Jazz.
So this is a silly question.
Yeah.
But why don't we just like put air into like the bone marrow so that it can transcribe theoretically everything into the bone marrow so that B cells aren't self-reactive?
I'm not sure we actually understand all of how AIR works well enough to do that, but I also don't know that we understand all of the details of this to know about other negative consequences. As you'll see when we get to peripheral tolerance, some of the peripheral tolerance mechanisms are pretty good on this, so it's not like we really need to fill that gap. This was described first in a paper like the paper you see here. So in here they were actually looking to see, they were looking at people. I think this is people. Can't remember if this one's people or this one's mice. I think this is people. They were looking at individuals who might have been people or mice because I can't remember, who did or who didn't have AIR. And they were looking to see which genes no longer got transcribed in the thymus. And so they could find that AIR seems to control transcription of a lot of things that are usually only in one specific tissue. And so AIR was regulating how the thymus made all of those things. So AIR allows the thymus to make pre-proinsulin so that T cells can be tested against insulin. AIR allows the thymus to make pre-pro-neuropeptide Y, and all of these other types of things that might be found only in one tissue. AIR was first described in a group of patients who don't have it. So basically what happened is there are some people who don't have AIR, and people were like, what's wrong with these patients? We can't figure it out. And when they did a study, they found they all had a mutation in AIR. That's how AIR was described. These patients have a disease called APSED. I will say what APSED stands for just because I can. No, you don't have to remember it. But APSED stands for autoimmune polyendocrinopathy and candidiasis with ectodermal dystrophy. The idea with AIR patients is that AIR patients have multiple autoimmune diseases at once. Because it's not that one T cell slips through the cracks. It's like all the T cells slip through the cracks. And so you can see, if you look at a group of APSED patients, they all have like a different combo of autoimmune diseases. So you can see 85% of them have hypoparathyroidism, 72% of them have adrenal failure, 60% of the women have ovarian failure, 18% have diabetes, 14% of the males have testicular atrophy, and on and on and on. And so basically these patients have multiple autoimmune diseases at the time because their central tolerance for T cells is so messed up because they can't do selection of tissue-specific antigens in the bone marrow. Weirdly enough, they all have recurrent yeast infections in the mouth, which is candidiasis. They also have something weird going on with their fingernails. So you can see this is the fingernails. They've got a bunch of things that we don't entirely understand what's going on. In mice that don't have air, their salivary glands are a disaster. It seems like they have this crazy autoimmune attack on the salivary gland. So this is sort of the good side of T cell development. But we also have a bad side of T cell development. I'm going to just like briefly say it, and I'll say more about it next time because, like I said, I want to leave you on a cliffhanger for your weekend. Every single T cell in your body had to be positively selected in the thymus, right? Every T cell in your blood, every T cell that's in your periphery got a pat on the head and was told, you're doing a good job. You're allowed to leave the thymus. You get positively selected. What did the T cell have to do to get that pat on the head? What had to happen for that pat on the head to happen? And for that T cell to be allowed to leave the thymus? Yeah, Emily.
Just any old medium pat.
It has to, so it had to get any old medium pat. What does that mean that it had to happen? It bound to something. What's in the thymus for it to bind to? Lots of things. What are those things? Proteins? Are they foreign? No, they're self. That means every T cell in your body has to bind to self a little bit. It could not have made it out of the thymus if it could not get a weak signal from self. Every single T cell in your body is a little bit autoreactive. It had to be able to get a low signal in the thymus to be positively selected and be allowed to leave. So every single T cell in your body is a little bit autoimmune. You got rid of the ones that were high autoimmune, but every single one that got a pat on the head in the thymus got a pat on the head because it bound self. So every T cell in your body is a little bit autoimmune. The way our immune system basically has evolved, we are like right standing on the edge of autoimmunity all the time. And it's only because we have peripheral tolerance that it protects us from falling over that edge. And so you can see that in T cell development. I will see you and we will talk about a little bit more T cell development, and then we will finally be in the periphery on Monday.